Hello friends, this video is on neuromuscular blocking drugs or skeletal muscle relaxants. This video will be useful for first prof students where a reasoning question will be asked. Succinylcholine is a drug that causes depolarization and yet causes muscle relaxation. Explain why. Or it could be important for second year students where we will discuss the pharmacology of succinylcholine, the depolarizing blockers. And of course for neetpg or PG entrance exams. So let's have a look at this particular topic. Skeletal muscle relaxants or neuromuscular blocking drugs means they block the transmission at the neuromuscular junction and thereby they bring about muscle relaxation. Now skeletal muscle relaxants or almost they cause muscle paralysis. When do we require these drugs? They are required before any surgical procedure. I mean, during the surgery, the patient's limbs should not move. It will interfere with the surgical procedure. So therefore, we need to give skeletal muscle relaxants that will completely relax or paralyze the muscles. Or before certain short procedures like endotracheal intubation. So these drugs are given. So these are neuromuscular blocking agents. Therefore, first let's see the physiology of neuromuscular transmission. Quick recall of the neuromuscular transmission. You know, at the neuromuscular junction, there is release of acetylcholine. Now, this acetylcholine binds with its receptor, the nicotinic acetylcholine receptor on the motor end plate. And this binding will open the non-specific cation channel. Influx of cation will create end plate potential or end plate depolarization, and then it will convert into an action potential which will propagate into the muscle, and as a result the muscle will contract. So this is how the neuromuscular transmission results in the muscle contraction, and the drugs that block the neuromuscular transmission will cause muscle to relax completely, and that is what we are looking at. Now basically before we start, I need to highlight 2 points here. First is about the cholinesterase enzyme. Now, once acetylcholine is released and it binds with the receptor, and then it creates that end plate potential or end plate depolarization, immediately its action is terminated by the enzyme called as cholinesterase, also called as acetylcholinesterase. It causes hydrolysis of acetylcholine and the action of acetylcholine will be terminated. Now, there are two types of cholinesterase enzymes. One which is present here, it's a true cholinesterase enzyme which is present in the neuromuscular junctions. The other one is called as pseudocholinesterase enzyme or butyrylcholinesterase enzyme. Now, this enzyme, pseudocholinesterase, is synthesized by the liver and it is present in the plasma. It comes into the plasma. So it's not present at the neuromuscular junction. The pseudocholinesterase enzyme is present in the plasma. That's point number one. And the second important aspect is the channel which is associated with the nicotinic acetylcholine receptor. This is a non-specific cation channel. It creates that end plate current or end plate depolarization. But further action potential has to propagate into the muscle, and for that we also have voltage gated sodium channel or NAV, and this will cause action potential generation and propagation of action potential into the muscle, and it will result in the muscle contraction. So difference between this channel, the nicotinic acetylcholine receptor associated channel, and the voltage gated sodium channel. So these 2 points being highlighted. Now let us talk about the neuromuscular blocking drugs. Now basically there are two types of neuromuscular blocking drugs which result in muscle relaxation. One, competitive blockers. They compete with acetylcholine for binding with the nicotinic acetylcholine receptor, and therefore the acetylcholine cannot bind with its receptor, and therefore the channel cannot open, and therefore muscle cannot excite. Muscle will remain in a relaxed state. D-tubocurarine or curare derivatives, they belong to this category, competitive blockers. Now that category is easy to understand. Their pharmacology is easy to understand. So our focus is going to be the other category, which is non-competitive blockers. Non-competitive blockers, also called as depolarizing blockers. That is, they cause persistent partial depolarization of the muscle membrane, and thereby they do not allow the muscle to excite further and contract anymore. That is how they will bring about muscle relaxation. And this is where students had doubt. If the depolarization leads to muscle contraction, which is a known fact, then how come this drug is causing depolarization of the muscle, but muscle is relaxing? Let's understand this. Now, the category depolarizing blocker or non-competitive blocker, the prototype drug is succinylcholine. What is succinylcholine? It's two molecules of acetylcholine. Two molecules of acetylcholine linked end to end. So 2 acetylcholine is equal to 1 succinylcholine. You can remember like that. And it's a neuromuscular blocking drug given before surgery or procedures like endotracheal intubation. We have done that already, and it causes skeletal muscle relaxation. Now, it has got extremely short duration of action. It can be generally given for short procedures. It has extremely short duration of action. There is rapid hydrolysis of this drug by the enzyme pseudocholinesterase. Look, you will give succinylcholine by parenteral route, not by oral route. And as you give it by parenteral route and it reaches the plasma, in the plasma there is this enzyme called as pseudocholinesterase. And this enzyme will rapidly hydrolyze succinylcholine so that only a small amount of succinylcholine will actually reach the neuromuscular junction. That's point number 1. And true cholinesterase, which is present in the neuromuscular junction, does not have the action on the succinylcholine. However, it's still very small amount of succinylcholine is good enough to cause the skeletal muscle relaxation. We will see that later. Just an additional point. This point I often call it physio plus plus or pharmac plus plus. In this case, if you write it in the exam, you get extra marks. Genetically abnormal variant of pseudocholinesterase. The enzyme pseudocholinesterase, which is present in the plasma, there are certain genetically abnormal variants that are possible in some individuals. Succinylcholine action of neuromuscular blockade, it will get prolonged because of these genetically abnormal variants of pseudocholinesterase. How do you know that a person might have genetically variant pseudocholinesterase? There is a test called as Debbucane number test. Debbucane is a drug that is given, and it may identify the action or rather it may identify the pseudocholinesterase enzyme, whether it is normal or variant, by the prolonged action of succinylcholine, where this enzyme will not act properly. All right, now let's understand the mechanism of action of succinylcholine. Here in this diagram, we can see that acetylcholine is released at the neuromuscular junction. Then acetylcholine binds with its receptor, and then once it initiates the end plate potential, end plate depolarization, then there is sodium channel which is activated, and further depolarization and action potential will propagate in the entire motor units, all the muscle fibers which are associated with this. So that's the action of acetylcholine. You can see here, if there is no acetylcholine present, what is the status of the channel? If no acetylcholine is binding with the receptor portion, the channel remains in a closed state. And with succinylcholine, what really happens is succinylcholine binds with the receptor, the acetylcholine receptor, and it opens the channel. So action is similar to acetylcholine but slightly different. As we have said that acetylcholine is immediately hydrolyzed by the true cholinesterase present in the neuromuscular junction, but that enzyme does not have that action on the succinylcholine. So succinylcholine, once it binds with the receptor of the acetylcholine, then it will cause persistent opening of this channel which is associated with the receptor. Opening of this channel will cause persistent end plate potential. There will be a cation influx and persistent end plate potential. End plate will remain in a depolarized state for a long time. And then it means initially a few depolarizations will be completed and will travel into the muscle. Initially, that end plate potential will convert into the action potential, and those sodium channels which are involved, they will cause those action potentials to be completed and travel into the muscle, so that initially there will be first few muscle contractions. Those are called as fasciculations, muscle fasciculations. Some isolated twitches, isolated muscle contractions will be observed immediately after you give succinylcholine. But then what really happens is this. Sodium channels, they have three states. When no excitation is happening, they are in the resting state. Then after binding of acetylcholine, and then there is end plate current, then sodium channels are activated, they are open, and after that they go into inactivated state. Please remember this. Sodium channels are initially activated when succinylcholine binds with the receptor. So initially a first few action potentials will be completed, but then the sodium channels are thrown into inactivated state. Now if they have to be activated again, from inactivated state they have to go to their original resting state, which does not happen. Sodium channels, after initial few action potentials were completed, they were thrown into inactivated state, and now the motor end plate is partially depolarized, and in that potential the sodium channels will remain in an inactivated state. So now the sodium channels cannot open because they have not gone back to their original resting state. So they cannot be opened again, and action potential cannot be generated again. So initially first few action potentials are completed, but sodium channels are thrown into inactivated state, and it's then difficult to generate the action potentials any further. So that's what happens here. Succinylcholine, when you give it, initially it will cause the action potentials to be completed. Let's have a quick summary. Mechanism of action. It is an agonist at the nicotinic acetylcholine receptor. I hope you can see this or read it. Persistent depolarization of the motor end plate. At the motor end plate, it causes a persistent depolarization. Now, succinylcholine block has been described in two phases. If it's a short duration, small dose of succinylcholine, there will be a phase 1 block. If it's a continuous exposure of succinylcholine, then it will go into a phase 2 block. The result is same: muscle relaxation, muscle paralysis. Let's see how it happens. Phase 1 block is called as depolarizing block, which we saw just now. Depolarization at the motor end plate, it happens initially. It spreads to the adjacent membranes. Yes, there are those voltage gated sodium channels, so it spreads. And then contractions of motor units. So initially you will see some fasciculations, some muscle contractions will be visible. But then succinylcholine remains bound to the receptor, as it is not hydrolyzed by that acetylcholinesterase or true cholinesterase. So succinylcholine remains bound to the receptor, and its action is something like this. Diagrammatically you can see here, succinylcholine is bound to the receptor portion where acetylcholine was supposed to bind, and it does not allow this channel, the associated channel, to close. So the channel will remain open, and there will be a partial depolarization at the motor end plate because of a cationic influx followed by some cationic efflux. So partial depolarization at the motor end plate. Look, if the muscle has to contract and it has to generate some tension, after excitation, after depolarization, it has to repolarize also. Motor end plate should repolarize in order to depolarize again. But imagine that it is in a persistent partial depolarized state. So there is no recovery, no repolarization, no further depolarization therefore, and therefore no muscle tension being generated. So after initial contractions, muscle fails to contract, it will relax completely. It cannot excite completely, it cannot contract, it relaxes. So skeletal muscle, as you can see here, channel remains open, so persistent depolarization at the motor end plate. No repolarization of the motor end plate, so no further excitation possible. I mean, for muscle to excite, it has to first recover from the previous excitation. No repolarization. So, and we said already that the sodium channels have been thrown into inactivated state now. Hence, even if impulse arrives, imagine that the nerve is sending some impulse to the muscles. Even if the impulse arrives at the neuromuscular junctions and acetylcholine is released, yet no excitation can travel into the muscle. Why? Because those voltage gated sodium channels, they are in an inactivated state. They could not return to their original resting state so that they can be excited again. That's not happening, and therefore no contraction possible, and results in flaccid paralysis of the muscle. So that's the key factor that we are seeing here, that sodium channels after initial excitations, after giving succinylcholine, initially excitation did happen, some contractions happened, so muscle fasciculations were visible, but sodium channels went into their third state. As they have a cycle to complete, they went into inactivated state. Now unless they go back to resting state, they cannot be excited again, and that is exactly what is happening here with the succinylcholine. All right, let's move on. Now, talking about the phase 2 block, we just saw in the phase 1 block, there was persistent partial depolarization because that channel associated with nicotinic receptor, it is not allowed to close. So persistent influx of cations and therefore persistent depolarization at the motor end plate. That was phase 1 block, depolarizing block. Phase 2 block is called as desensitization block. This happens when there is prolonged exposure. If the succinylcholine is given repeatedly and there is a prolonged exposure to succinylcholine, I mean succinylcholine levels are there, persistent. So what happens is, now the end plate depolarization will wane. Slowly the end plate depolarization will wane and repolarization will happen, membrane will repolarize. But now there is another problem. Receptors become desensitized. Now those receptors will become desensitized to the acetylcholine, and therefore the block will happen something like this. If you can see in the diagram, that succinylcholine is persistently bound, in a persistent manner it is bound to the receptor, and acetylcholine just cannot displace it, and therefore now the channel remains closed. Now this is called as the desensitization block. Acetylcholine action cannot happen now. This block is similar to the competitive blocking drugs. We said that there is another type of block called as competitive blockers, D-tubocurarine. They also produce the similar block. They go and bind with the receptor portion. Now the acetylcholine cannot bind and cannot open the channel. So similar type of block also happens with the succinylcholine, but in the phase 2, which is called as desensitization block. And let's have a summary of this neuromuscular blockade in a diagrammatic manner. Okay, so acetylcholine, it binds with the receptor portion and the channel opens. muscle excites and muscle contracts. This is how things happen normally. Now the second is competitive blockers. How they block? They bind with the receptor where acetylcholine is supposed to bind, and then they shut the channel. Channel is not allowed to open at all. Acetylcholine action cannot happen. Acetylcholine cannot bind, so channel cannot open. And the third one is succinylcholine. When succinylcholine binds, it does not allow the channel to close, and therefore persistent depolarization. So just compare second and third diagram. The curare or D-tubercularin, it does not allow the channel to open by the action of acetylcholine because it is sitting where acetylcholine was supposed to sit, bind. And succinylcholine, when it binds where acetylcholine is supposed to bind, it does not allow the channel to close, causing persistent depolarization and sodium channel inactivation after initial few action potentials completed. That's the important point to be highlighted. After initial few contractions, now the muscle will stop contracting, it will relax completely, and that's how the skeletal muscle relaxants work. We discuss many such topics on a regular basis, so it will be a good idea to subscribe to the channel.